Welcome back. We are going to hit fluorescence activated cell sorting. This is a form of flow cytometry. It's a special type. Clearly the word fluorescence activated means that there has to be fluorescent molecules. And we've seen with antibodies that you can tag, i.e. chemically attach molecules that will release energy in certain wavelengths that we perceive as color, and that's called fluorescence. This does separate cells into subpopulations based on this fluorescent labeling. It is said to be sorting, and it's a little more complex than just the general flow cytometry. Cells must be introduced and stained with fluorophore conjugated antibodies. So the term is fluorophore, which can be separated from one another depending on what the fluor is that they've been stained with. This is the 2-stain, 3-stain concept we talked about. So a cell that expresses one cell marker may be detected using an FITC conjugated antibody. And when they say the word marker, they're most likely referring to proteins on the cell surface. There are some lipids and carbohydrates that may also have antibodies made to them. So most likely it's proteins, but it could be other biochemical molecules. And so if you have a certain cell marker, then that cell would fluoresce green because FITC is a green wavelength. And another cell type in that mix. So again, we've got a mix of cells here, a mix of cells. So another type in that mix is maybe has a different protein on its surface or a different lipid or carbohydrate. And that will be detected using a phytourethrin conjugated antibody. That is a red wavelength fluor and specific for that marker. So we could find cells in this mix that do not have either markers, so they will not have green or red. We can find cells that have both, and they would be red and green, or probably yellow if you overlapped the channels. Then you've got the green only and the red only. So again, you've got the 4 different cell types that could be expressed. And so that's the basic task of this type of flow cytometry, is to sort the cells out. These cells can be then cell cultured, grown up, frozen, studied, etc. at that point. So there's other experiments, again, under diseases, different organs, different conditions. One is able to study. Again, you have a population of cells. On the right is the typical machine. It's a little out of date there. That screen looks ancient. But anyway, there's the lasers and the recorders and these kind of things, deflection plates, and there's actual place where cell collectors are. It's not so apparent. But anyway, all that material is there, and when you look at a cross section of that, you have your cell suspension, a mix of cells, and some type of fluid that is added, buffer, something that these cells will behave as normally as they can. Notice they've got red and blue here. Okay, so they've got red and blue cells, whatever those are. And the nozzle has an ultrasonic nozzle vibration mechanism, and so that shakes the cells that may be attached up, and this pore at this point right above where the laser is having it interrogates the cells as they drop. One cell at a time in one drop comes through. So the laser hits it, and whether it's red or blue or red or green, we have detectors, and we then analyze exactly what went through. So there's a keeping track. If you ask the question, how many cells in the suspension, you'll count the number of cells and how many were red, how many were blue. And then small groups of drops uh, uh, right here eventually can get a negative charge or a positive charge. And so there is a charge that can be applied right after or during the detection. And so then they have a deflector plate, and this on one side pulls the negatively charged molecules and deflects them to one side, and there's a collector here, so that all the blue. There's then those that have a positive would be potentially moving into this cell collector. Cells that might have both or undeflected could be in this undeflected flask. So here's this sorting mechanism based on electrostatic charge that's applied in this process. Okay, so this is maybe a little bit another way to look at it. Here's your laser, here's the nozzle tip, you're shaking into drops. And so individual cells are interrogated by the lasers that are coming through. And so you get one drop at a time. Each drop is, there's an electrical static charge that's given, and it's dependent on the fluorescence. So when the laser says green, then the machine will give it, say, a positive charge. If it's red, it will be given a negative charge. So there is some software that must read almost instantaneously what the marker is on the cells and give them a charge accordingly. And then we have deflector plates to where one charge, here's the green, and then here's the red depending on the charge and the deflection here. So deflection plates will either attract or repel the cells depending on the nature of the deflection plates. So a single FITC green stained cell, this means it has the marker for the antibody that's tagged with FITC, and it would be attracted to the right-hand side. So I guess right is not as we look at it, but as the machine is looking here. So this is what they're calling the right here. And then the red is not deflected, and it ends up being collected at this point, and just droplets with no cells or undeflected cells would flow through here, and I assume there's some type of container there. So these collections of cells can be then cultured. So then each one of these is cultured, and you have new cells that now can be studied, okay, depending on the property. So if you took a tumor, shook it up, and you stained for two major cancer proteins, you could divide the tumor cells into those that had a lot of whatever the green is detecting or the red, okay? And so you can further fine-tune exactly what the cells are within that tumor or condition or situation, okay? So fluorescence, activation, there's a charge here, deflection, collection, and then cell cultures at this point. Okay. And this may be some words. I believe it pretty much discusses what we've talked about. And just in talking about the vibration, the tips. So here are some cells, and it looks like they're stained with a certain dye. Looks like it's red here. So you have cells that are stained and cells that are not stained. Here's the laser. And label cells are sent, and there must be some type of charge to where the heavily dyed cells collect here. The CD, gamma Y, bright cells are heavily stained, and the ones that are weakly stained, I call those dim. If you looked at the nature, then you could figure out the amount of cells that were dim versus the amount that were bright. And so you can get a percentage of cells from your original culture here. And then these, of course, can be recultured after you sorted them, okay? After you sort them for further testing. So just remind you about the antibody and the area that will bind some antigen on the surface. And remember, the fluor is attached here, okay? So the fluorescent molecule, fluorescent fluor is right there, the green or the red, the FITC or whatnot. So just remind you a little bit. So I like this because it shows the fluorescent dye on the antibody that's bound to some receptor. So now this cell has 8 of these proteins. So in the mix, there are those that don't have the marker and those that do. So the red versus the dark black. Here's a detector. Okay, at this point, the FITC and the fluorescence and the granularity, so the complexity. There's an electrostatic charge given to the cells as if they have a red color. If they're not stained red, they will not be charged. So the deflector plates have a negative charge and a positive. And so the red is given a negative charge and it's deflected in this direction and captured here, the positive sample. And these are cells that are undeflected or collected without any red dye. So they don't have the markers on the surface of the cells. So cancer cells in a biopsy and non-cancer cells, whatever this could be. Here's another one just showing the antibodies, the detector, the deflection plate, and you're detecting fluorescent and non-fluorescent. Again, these can be cultured at this point and counted, and you get the numbers under a condition. So remember, we discussed poly and monoclonal for cancer treatments, and so we can detect then cells and the tumor cells and what they might have. This is just reminding you of all the different interactions that we can do here. Here's our antibody, monoclonal antibody, MAB. So there it is. And the MAB can have a radio-labeled ligand, so we can make this tumor cell have radiation that can be tracked in the body or in the mouse. Here is a killer cell that is detecting the antibody and bridges the gap, and the killer cell can give the apoptotic signal. Here's a liposome that actually has a attachment molecule to the tumor, and you can deliver whatever products, poisons, medicine, inside this lipid enclosed, kind of like a little lipid M&M here. Here's a connection where you have some type of drug that is made with this enzyme. So this enzyme attaches to the tumor. You add a prodrug, and only where their enzyme next to the cell will you get the drug that then can go in and maybe attack the cell. Here's a toxin that's been attached to the antibody to kill. Here's a growth factor or other kind of signaling molecule that's attached as well. So again, here's the cancer cell, and the cancer cells will have all sorts of proteins. Many of these will be abnormal and not in non-cancer cells. Okay, so this is just kind of an overall about what monoclonals could do. And of course, we've been talking about cell sorting here and floors, right? Here's the floors here at that point, okay? This is just a labeling protocol. If you're curious, you don't have to see that. Here's again the same thing, just showing the red, the green, and the unlabeled. Sometimes I have different, same slide several times. And here's the intensity. Here's the frequency in the mix, so you get an idea of how much are red, green, or no stained. Okay, so you can get an intensity. And again, it's how you read the data. Cells that are highly stained with green, high red, no red and green. Cells that are stained with neither. So that's how you can look into a mix of cells and figure out whatever these two markers are here. They're clearly not together. So you're able to efficiently, in this cartoon, 100% selection of the two colors and the two populations of cells. These are just some dyes that you use. You don't have to memorize this. And the wavelength of light and the different lasers that you need and the colors and the spectrum. Okay, here's another chart, just shows the colors, the emission spectrum, how you attach. So there's a lot of chemistry to this. Again, you can, here's the, where's the I don't know if it, yeah, there it is. So this is FITC right there. And this will be where the phytourethrin in this area, the red. Okay. Just wanted to remind you, in case you remember in genetics, where proteins can be tagged with the fluorescent protein that is different from a fluorescent antibody. In this, and then we'll talk a lot about this in molecular, but in this case, we took the histone gene, added the gene for a green fluorescent protein. We made a hybrid protein that then is inside cells, and then if we apply a fluorescent wavelength, we can see the nucleus because it's where the histones are. That is not facts. That is not anything we've done. I just wanted to remind you, in case you had seen some type of protein tagging before, that is completely separate from the fact. This is just a discussion of a machine. I think I found it somewhere, just showing that a lot of these machines have multiple lasers. So they deal with, I mean, when you sell a machine to labs, you better have all the possibilities. So that was the only big point there, if that was a big point. So here's cells that express antigens, which are unique to that cells. We have tagged antibodies. They have a fluorochrome, and then we can detect them. Here's the droplet formation, just showing kind of what we've seen before, but I just had some more pictures. Some information determined by flow cytometry, including facts. Some of this we've talked about, some we haven't. But if you do inquiries down the road and you need this kind of information in these scenarios, then certainly you could, you had enough background, hopefully, to, that you could deal with this. But just wanted to let you know. This is a diagnostic tool, and this is CD4 cells, which have a red, CD8 cells, which have a green, and here's double positives here, cells, okay, that have CD8 and 4. Okay, so patients that have HIV, the CD4 right here to CD8 ratio is monitored, and it should be 1.4 to 2. When the CD4 drops, so that means that there would not be these cells or very few, then you would have an indication of full-blown HIV. Okay, so it's a diagnostic tool here. There's other techniques as well, but that just shows you an example, and this just shows how we're another method where we can look at cells under different growth conditions, G1S, G2 mitosis, and you can look and see what cells are doing what at what time. And there's a correlation here, the double negatives. They're sitting in S a lot more than these other guys, which is interesting. Not sure what that means, but it is interesting data. So you can expand the facts into many different 12, 15 if you have a sophisticated enough, and you can compare. And so this is the original side scatter, forward scatter, just what the cells are looking like, side and complexity. Then here's CD4 to 8 story, CD24 to 4, CD3 to 4, CD24 to 8, 3 to 8, 3 to 24, and just the profile of the forward scatter. So right away you could tell, say, a blood sample, and you could stain that with all these different antibodies, and you could get a real interesting workup of a person looking at these different cells. So once, if there's a disease in the database, and so bioinformatically with enough patient samples, all these profiles, the percentages, what these look like, can all be in a database. And then when you have new samples, they can be compared and potentially speed up the diagnosis here process. This is some company was showing all the different possibilities they could. They have something called imaging flow cytometers, and they give you detailed image of every cell and even these rare subpopulations, and they're showing the different interactions and things they can do with this cell in suspension as they're going through the detector. They can get a snapshot of morphology, DNA damage, cell cycles, signaling, cell interactions, who's alive, who's dying, proteins inside together, co-localized or not. I thought that was really neat. So that's probably one of the newest machines that have beyond just the flow cytometer and then the facts. It's called an imaging flow cytometer. So they combine the characteristics, and they've added now sophisticated imaging, which you would predict that techniques change. So now this is something like, so this is the first time I've seen this, so it's probably a fairly new machine. Very interesting now, the workup you could do if some students had inquiries to solve down the road. This is just the glossary, okay? some new terms. So a lot of cell biology is hard because you've got to learn a new language. So I would say for studying, you definitely need to get note cards and make definitions. You need to then put together concepts, and then you probably need to make some essay questions based on this, and then have others read them and give them a thumbs up or a thumbs down. So that will prepare you for my test. And so you're saying, oh, well, this is a lot of writing, and yeah, cell biology is hard. Remember, chop the wood first, then you get the fire. So yes, a lot of new words, a lot of new concepts, put them together in an essay. Okay, so there you go. Dr. Bush, how to study. Do this. Well, Dr. Bush, I don't want to do that. Well, okay. But don't try to horse me and say, well, Dr. Bush, I made note cards for everything, and I did all the concept maps. You know what I'm going to ask you? Bring them then tomorrow and let me look at them, make sure you did it right. Uh-oh. Had a student do that. Uh-oh. Never saw them again. Uh-oh. So don't BS me. Thank you. So I hope you enjoyed facts analysis here. I think I'm getting close to my 2 hours from last week here. Woo woo. If I get some energy this Labor Day, I may move ahead and do some more. We got lots to do in this small little lecture, what we call lecture 2, which is techniques. Okay, enjoy your reading and being happy and perky. Okay, get ready, chop that wood. See you next time.